All right, let's get started. Welcome to a new initiative that Wipro has undertaken called regulatory briefs. And the first topic that we have under this initiative is what's new in MIDD. And I have great pleasure in welcoming Dr. Court Bradley to speak on this topic. MIDD, in my experience, the phrase MIDD is used as a noun, verb, and an adjective. Dr. John Croth Bradley is going to cover, I guess, all these three different parts of speech in her um, introduction background to MIDD as a practice as well as MIDD as a meeting mechanism with the US FDA. This webinar is being recorded and we invite your questions and comments. You should be able to type them in the chat box, and if I can get my technique correct, I may be able to see those questions as well to moderate the discussion. If you experience any technical difficulties, please feel free to reach out to Holly. She's standing by. And at the end, if you can take a minute or actually 15 seconds to provide feedback, it will be very helpful for us to plan the subsequent webinars. Now, about Dr. Court Bradley. She has briefly, I will summarize her background briefly. She has over 30 years of experience in drug development, ranging across multiple therapeutic areas: oncology, cardiology, anti infectives, rheumatology, and rare diseases. Her work included early as well as late development in several molecular diseases, including small molecules, therapeutic proteins, and ADCs. She published over 90 papers, and professionally, she's very active. She serves on the editorial board of Journal of Clinical Pharmacology and Clinical Pharmacology in Drug Development. And she earned her PhD from UT Austin and her PharmD from University of Minnesota. So let me welcome Dr. Court Bradley. The stage is yours.
Thank you very much for the opportunity to address this topic, and I look forward to the questions and discussion that we have. Just a brief outline. Because this is primarily a regulatory perspective, I'd like to start with a little bit of a review of drug development so that we're all kind of thinking on the same page. Spend a few moments talking about models, and then combining the two, model-informed drug development, and finally a brief discussion of dose optimization, which is the reason that we do drug development. So drug development, we begin with the end in mind. What is the unmet medical need that we're interested in providing a therapeutic option for? And who are the patients that will need this treatment? Then we think about, in addition to the indication, we think about the medication. What's the evidence that it's going to work? How much medicine is needed? Is it something that we'll give for a short course, like an anti-infective? Or is it something that's going to be lifelong treatment? How big is the dose? What's the right dose to give? Will everybody get the same dose? And how will it be administered? Orally, in a tablet, a capsule, a liquid, or parenterally, a subcutaneous or an intravenous administration? Drug development answers all of these questions. Now, in ancient times, sometime before 1962, In 1938, after a tragic disaster, a sulfa suspension that was used in the treatment of infections in children was was co-administered with a diluent that included the elements of antifreeze. And so many of the children died as a result or were poisoned as a result of taking this hopefully life-saving medicine. In response to that, in 1938, the Federal Food, Drug, and Cosmetics Act was introduced. And it increased the authority of the FDA to monitor drugs. Prior to that, basically, you just developed a medicine and sold it. Evidence of safety and quality were now required. So, evidence that the drug was somewhat safe in the doses that would be administered, and quality that the medicine could be manufactured in a safe, clean, and reproducible manner. Now, the FDA had 180 days to review the applications, but they could extend the time if they felt the NDA was incomplete. In 1957, thalidomide was a medicine that was introduced in the European market for the treatment of nausea and vomiting associated with pregnancy, sometimes called morning sickness. By 1960, the FDA application had been filed. But as we know from the story, Francis Kelsey, the medical reviewer, felt that there was inadequate information describing the safety of the medicine, and so did not agree, despite considerable pressure, that it should be approved. At the same time, the reports of the teratogenic effects, the failure to develop normal limbs in the babies that were born to moms who had taken it, were being reported. And so ultimately, the drug was withdrawn from the European market and never approved in the United States. Now, coincidental with all of this, we were entering the space age. 1957, the Sputnik was launched, and quickly the US worked to catch up. So science was also being applied to drug development, and there were congressional hearings, certainly in response to thalidomide, but independently, to better understand what evidence of efficacy would make sense. And in 1962, the Kefauver-Harris Act was passed, and it said that basically we have to demonstrate that safety and efficacy is required, in addition to quality, of course. I just want to consider two medicines as an example of how drug development was. Furosemide, Lasix, right now, even 2 generations after it was approved, is one of the most prescribed medicines in the United States and UK. Patented in 1959, it was approved in 1964. It's approved for the treatment of edema, hypertension, and the current USP dates from 2010. Similar to many older medicines, it does not include a clinical trials section. But if you look through the USPI, you can see that the safety and efficacy are not described in the usual clinical trial format, but rather they appear to be added on. So I can only imagine that the initial approval was simply the results of what the physician investigators who had used the medicine reported. In terms of pharmacokinetic studies, there are 2 in the label, an absolute bio study and geriatric PK. There are no models mentioned at all. Now let's contrast Lipitor, atorvastatin. In 2023, it is the most frequently prescribed medicine in both the US and the UK. It was patented in 1986 and approved in 1996. You can already see the impact of the Kefauver-Harris work because it took 10 years for the drug to be developed. It's approved for the prevention of myocardial infarcts, as well as, along with diet, to decrease the LDL cholesterol. The Section 14 includes clinical trials. And I draw to your attention that there are 17 placebo-controlled trials mentioned for safety. And there are 7 or 8, depending upon how you slice the pie, clinical trials mentioned for efficacy. In terms of pharmacokinetics, there's an absolute bioavailability study, food fast, 24 drug-drug interactions and the normal complement of special populations, geriatrics, pediatrics, hepatic, and renal impairment. The population PK model is mentioned in pediatric PK. Now, while Lasix was developed almost without clinical trials, Lipitor, I would argue, had too many. And so for efficient, effective drug development, I would argue that models allow us to jumpstart development. And there are ways that we can answer questions without having to study absolutely every different type of combination of interest. We can do better than 17 placebo-controlled trials. So what are models? They're basically a three-dimensional representation, typically on a small scale. You can have model airplanes, you can have model cars. If you're interested in updating your landscape, you can even buy little trees and little plants and move them around. Much easier to work in a smaller scale than the original. Now, in drug development, it's an abstract description of a concrete system using mathematical concepts and language. And just an example, y equals mx plus b, one of the simplest models, the equation that describes a straight line. And here we have a study that was conducted. A group of students reported how long they studied and what grade they received. And when you fit a straight line through these data, you solve for the fact that their score on the exam, the y, is equal to 4.6 times the number of hours they studied, x, plus 52. Well, how to interpret that? We just say that for every additional hour studied, that their score would increase by about 4.6 points. And even if they didn't study at all, the y-intercept predicts that they would get a score of 52. Now, this is a pretty exhaustive study. They have collected all sorts of data. If we only had this data point and that data point, we wouldn't need to do all of this, collect all of that data. It would be much straighter and easier. Now, I will argue that every model that we can implement, and there's tons of them in drug development, is simply a more complicated example of this. When we work with models, the process is basically the same. We collect the data that we think are of interest, in this case, test scores and amount of studying. But we've got lots of other examples in drug development. We collect the data, we fit the model to the data, we verify that we've got the right model, we evaluate the model. Does it answer the question that we're interested in, or is something else? Do we need to collect other data? And then validate that the model is correct. And there are lots and lots of different methodologies and experts on this area. And I'll leave that for another time. We use the model then to simulate the conditions not studied. As I mentioned before, that exam studying versus score in the quiz, we could have simply measured the lowest and the top, and we would have been able to explain the results in a similar way. Two things to keep in mind whenever one's working with a model and then simulating is interpolation. That means that we are answering questions within the conditions of the range studied. We had students studied between 1 hour and 10 hours. So we could answer the question, well, what would the score be predicted if a student studied for 6 hours? Alternatively, there's extrapolation, and that's when we try to make predictions about conditions outside the range. What would happen if a student had studied for 20 hours or 30 hours? We could predict that they would get 100% on the test, but we don't know that. We didn't actually study that condition. Something to think about and remember is that whenever you come across a simulation, you're reading a paper or you're looking at an advertisement, and they talk about a simulation, there is a model underneath that. And sometimes in order to better understand the impact of the simulation, it is helpful to dig deep into the model. Now, some of the models that are used in drug development, formulation and manufacturing, we're interested in studying dissolution and how different diluents will affect dissolution. We can assess stability, look at 6-month, 12-month, 36-month stability under a variety of conditions. We are interested in a lot of clinical and non-clinical data, pharmacokinetic models, PK/PD models, exposure response models for both safety and efficacy. Physiologically based pharmacokinetic models have been used a lot for evaluating drug-drug interactions, and um, quantitative systems pharmacology and drug disease models. How does disease progress when treated or without? And so on. There's all sorts of models. I'll show here one example. This is a stability model that was used for rituximab. Rituximab's a biological protein, and so the individuals were interested, the same types of conditions, can we use those? Are the same types of assessments that we use for small molecules predictive of long-term stability in proteins? Well, I think anybody could have told you that 45 degrees, which is the red line, is not good for a therapeutic protein. But let's look at 5 degrees or room temperature, 25 degrees. So they measured the amount of aggregates or breakdown, evidence of breakdown. They also looked at potency over a 6-month trial. They took these results and then they said, well, do these 6-month results predict what we would observe in over 36 months? So they've just taken the information for 5 degrees C and shown that in some cases, it's not a bad prediction. Sometimes it's an excellent prediction, and sometimes the model doesn't fit as well. But nonetheless, this is an example of the type of non-clinical or stability data that could be used and models developed and simulations performed. Pharmacokinetic models, of course, we do that all the time. Concentration time profile, this is indeed furosemide, Lasix, and looking at the concentration time profile in a group of women and a group of men. And this is comparing a generic versus the brand. Population pharmacokinetic model. This was a study that was involved in the administration of growth hormone to a group of children, and the doses, different doses, 0.25, 0.48, 0.66 milligrams per kg. And these data were collected, a sparse data sample over 5 years. So again, models can be used to answer a lot of really interesting questions. So if I haven't convinced you already, models help us have a better understanding. If we look at something and we see that there's no impact, then maybe it's not an important covariate. Or if we see that there's things jumping around and it doesn't explain it, then we need to ask other questions. What are other things that could be explaining? So we better understanding of the phenomenon or question, as well as better study design. And again, always focusing on the drug, optimal storage, stability conditions, what's the best dose, patients, exposure response, and interpersonal variability, observations, in terms of efficacy, tolerability, and safety. So again, models allow us to take the data that we have and put it together in an informative way that helps us do a better job and not require individual assessment at each time. A concentration time profile means that we don't have to measure all of the concentrations at every moment in time. We can take them at informative times and answer the same question. Now, models are not a free lunch. They are a tool, and there are things that we have to pay attention to. Prerequisites. One can't model if one doesn't have data. Occasionally, I've sat in meetings and people just say, you can model that. No, I can't. I don't have data. I need some data. Now, we can borrow it from the literature and other places like that, but ultimately, some data are required to be modeled. And the other thing that sometimes we forget is that it takes time for analysis, modeling, and validation. Sometime in ancient history, I was tasked with 3 population pharmacokinetic models that were collected. It was a drug that had 3 compartments. It had an active metabolite, and the data were collected over 3 months in patients that were quite ill. So I had a lot of dropouts. It took me an awfully long time to clean those data, a long time to analyze it. And unfortunately, at the time, the company didn't have quite the infrastructure that I needed. I needed special permission to run the computer programs longer than the timeouts that were already part of company policy. So I think that you can see that we have to make sure that we've got the data, make sure that we've got the time in order to do the modeling work that needs to be done. It's sometimes a challenge to share the interpretation with both the clinical team, the senior leadership, and others. Not uncommonly, someone will say, but you're answering a condition that didn't occur. That's right. We have to then take a few moments to explain the model. What are the assumptions and what is the learning from the model? Documentation. Again, those very first population PK analyses that I performed, I had piles and piles and piles of printout. Well, how did I share that with people? What amount of documentation was appropriate to put into a population report? Regulatory assessment. What was it? In those early days, the FDA had not yet published their guidance. What was it that they needed in order to look over my shoulder and come to the same conclusions that I did, or the most appropriate conclusions? And then finally, what's the best way to share knowledge? I had a conversation many years ago with a FDA reviewer, and I said, I've gone to all this work to do this population analysis, and there's nothing in the USPI. I need to have, if it's informative information that is answering questions of interest to clinicians, to patients, we have to have a way of putting them in the USPI. And similarly, publications. What's the best way to put data from a model into a report that is of interest to people who may not be intimately aware of all of the ins and outs of modeling? Now, because of these challenges, model-informed drug development is an organized framework that the FDA, the EMA, and certainly the ICH are working to address the issues of use of models so that if one conducts a model, it is acceptable internationally. ICH M15 is a working group that we anticipate their final results 2025, but they are working very hard to evaluate and bring together common language to describe how to do the best to basically supplement the population PK guidances that we have. To have one for exposure response, to have one for quantitative assistance pharmacology, for all of the important tools associated with modeling in drug development so that we can proceed to use these powerful tools in a very efficient manner, or at least not get caught up that one regulatory authority will interpret the results in one way and another will require totally different documentation. So now, in addition to the work of ICH and different groups developing guidances, there's another program, and that's the MIDD Paired Meeting Program that was initiated in 2018 and has been renewed in 2023 as a result of the most recent PDUFA. What the MIDD Paired Meeting is an opportunity to meet with the FDA to discuss some aspect, some question of model-informed drug development that is specific to a particular asset that answers an important question. Now, they published a couple of papers on the pilot results from the 2018 to 2022 program, and the most common questions were population PK, exposure response, and PK/PD. These three different types of questions made up over 75% of the 62 different assets that were reviewed. Now, oncology and neurology were the most common, but there were 14 different therapeutic areas participating. Under the current program, they are accepting quarterly applications, and I get a sense that it's approximately twice as big. So instead of one or two applications being accepted per quarter, maybe 3 or 4 will be accepted. The focus area are dose selection, clinical trial simulation, predictive and mechanistic safety evaluation. That's not to say that if one has a question of another type beyond this, but these are the ones that the agency has said they would like to focus on. So MIDD good practices. I don't think we quite can say best practices, but a good practice is to begin with the end in mind and to have a development plan. This will be our roadmap for developing. What's the target product profile? Is it going to be global? What are the studies that are needed in order to populate this product profile, including the prerequisite studies? So if one wants to study children, for example, what data are needed in adults before we study them? And we discuss with our senior company leadership and regulatory authorities. Is our senior company leadership on board in terms of the time and the money? Are the regulatory authorities on board with, we've mapped out a reasonable plan? They'll, of course, always say, it's a review question, but are we going in the right direction? Now, to supplement a clinical development plan or a development plan, an MIDD development plan is needed, where you identify the opportunities to incorporate models into the development plan, including the activities that are needed to support registration. Again, check with senior company leadership and regulatory authorities that this plan will get you to where you want, that you will be able to base important decisions on the results of modeling and simulation activities. And always important, because there's so many assumptions that are made in modeling, that we want to incorporate checkpoints to revisit the initial assumptions and results. It's not unreasonable, for example, to have a checkpoint after the first in-human study to make sure that the pharmacokinetics are doing what you think they should, and that the model that's developed to predict the pharmacokinetics will cover the range anticipated to be used in phase 2 and phase 3, for example. I'll just take a moment to step aside from MIDD to talk a little bit about dose optimization. Now, I think that's the reason we do MIDD. Dose optimization is our ultimate goal. You may remember that concentration of drug in plasma or dose and the percent of individuals responding. This is some marker of response. As we increase the dose, we anticipate that the number of individuals responding will increase, and ultimately, everybody who's going to respond will respond. We also know that evidence of toxicity occurs, and that as doses increase, there'll be some marker of toxicity or lack of tolerability that will occur. Now, again, you've been paying attention. You know this is model. There's a model, an equation you can fit on this line. Unfortunately, however, it's never quite this neat and easy. You'll see at the bottom, some people are not responding. Can we collect data in those individuals? We have a therapeutic window we have to describe. What's kind of the minimum amount of efficacy that's worthwhile? 100%. Then we also look at the toxicity. How much toxicity is too much? And so again, we want to describe the therapeutic index. I would argue that this model is the one that we are particularly interested in. So the key questions, how do we answer that? As I mentioned before, we have to be able to define that y-axis. What is the measure of safety and tolerability? Is it nausea and vomiting? Is it a blood count or something like that? How is efficacy defined? Is it growth rate? Is it the clinical response to an infection? That y-axis has got to be well described. What are the doses or concentrations associated with it? How is that x-axis described? Is there a biomarker or drug concentration measurement that needs to be opted? We translate, obviously, non-clinical to clinical simulations as much as possible. One doesn't want to run an entire drug development in a non-clinical sphere, but if data can be collected to put together that preliminary model of efficacy and safety and how they increase with increasing dose, we want to be able to then tweak that to move it from a non-clinical to a clinical environment. With clinical, we obviously have phase 1 studies, and that's where we have the broadest exposure typically. Phase 2, there's a more pristine group of patients than perhaps were included in phase 1. And in phase 3, we typically are confirming. So the dose range is much narrower, but the patients will hopefully respond or not, and we'll again be able to complete that initial concentration effect relationship. Now, just to kind of focus on what has changed since ancient history, we have placebo-controlled trials. I suspect that those early studies in Lasix were not placebo-controlled. They were open label, patients received Lasix, and they measured their clinical outcome. We also know that placebo-controlled trials are not always appropriate for infections or oncology or for conditions where we need to treat a child who is suffering from growth hormone deficiency. They need to receive something because they have a very narrow gap of how long they can respond. So then we have active comparator trials. We, at one point in time, we dosed to maximum tolerated dose. That is perhaps less acceptable now because we understand that what's tolerated in one individual may not be tolerated in others. And so we try to understand kind of what is the minimum effective dose, what's the maximum dose that can be reasonably administered for the population we anticipate taking it. More and more important are patient outcome studies. What is the patient's perspective on safety and efficacy? Again, in all cases, we collect data, we fit a model to the data, and we answer questions about what would happen under the conditions that perhaps were not included in the clinical trial. A systematic review of all information is important for what is the actual optimal dose. And there's a really nice discussion of this in the guidance that was published in 2023. Even if you're not developing an oncology drug, this is helpful to think about how do we know we've got the right dose. So now skipping back to MIDD, you've been successful. You've put in a request that you are interested in getting specific feedback, and the agency has said, yes, we would like to talk to you. This is a paired meeting, so you get 2 for the price of 1. One can either have a chronological development of the question or a deep dive. Perhaps the model is so complicated that one needs to have an initial meeting, initial work, and then a subsequent model to further understand the question. Or the 2 meetings can be on different topics. Now, the background document is not a core dump. One wants to have the relevant information thoughtfully summarized. Now, if you look at what are all the possibilities of things you can talk about, I recommend taking a look, again, even if you're not working in oncology, at the Oncology Dosing Toolkit. One looks at translational evidence. So what do you know in non-clinical circumstances that helps you understand in the clinic? What are the PK characteristics? What's evidence of safety, efficacy, and dose response? And even if you don't have the answers, the agency will be interested in what your thoughts are for answering these questions. So in summary, dose optimization in patients is the ultimate goal of drug development, and I would argue the ultimate model. Drug development plans, including plan modeling activity, facilitate clear communication of the assumptions, key milestones, and timelines amongst team members, amongst vertically throughout an organization with senior leaders, as well as with regulatory authorities, thought leaders, and other individuals who are helping us with our drug development plans. MIDD can be applied to quality, non-clinical, clinical, marketing, real-world evidence, and this offers a boost to the efficiency of drug development. So I thank you very much for your kind attention. And now I pass it back to Joga. Here are some additional things if you're interested in reading.
Thank you.
Joga, you're muted.
Can you hear me?
Yes.
Okay, something happened, I guess. Okay, sorry. So what I was saying is, let's see if the attendees have any questions, and I I can also start while we wait for the attendees to pose their questions in the chat. Um, so the question is, will the slides be available? I think the recording will be available to all. Um, so yes, you will be able to access the webinar material after the webinar. So while we wait for uh, folks to chime in with questions, let me start the discussion by asking um, Joan, given your extensive experience in pharmaceutical companies, for a for a pharmaceutical company to engage with a regulatory agency such as US FDA in the MIDD meeting, um, you know, in availing the MIDD meeting pathway, what would be a typical process internally?
Again, hopefully, a company, a sponsor would have a development plan, and they would have thought about what are the opportunities for model-informed drug development. Do they want a particular population PK? They've got the label in mind that they want to achieve, and they've understood where they can get those data from. But, as everybody knows, it's never a really clear cut response. There's always a few things that people have questions for. And so a company, again, I think it makes excellent sense to avail themselves of this particular tool that the regulatory authority are making available. Too often, I think a population PK analysis or an exposure response analysis is lost in the other meetings that we're having, our end of phase 2 or a type C meeting where another public health principle is being discussed. So I think if possible, companies should avail themselves of this, put together their question, try to do the best job they can in pulling all of the data together in order to ask a question that will be helpful in terms of moving their asset further along in an efficient way. And perhaps they can take they may not need to do a study. They may be able to identify a modeling and simulation opportunity that, again, collectively, put together properly, will just be huge in terms of advancing their program. Always a good idea to take advantage of this opportunity.
Thank you. There is a question in the chat about uh, is there a timetable to have the meetings with FDA after company request is accepted? Um, I can tag team with Joan in participating in this discussion. So I posted a link in the chat. I'm not sure if the attendees can see the link. Holly will have to address that. But to answer the question, yes, there is a timetable. Right now, MIDD meetings are accepted on a quarterly basis, and there is a timeline for when before which you should submit meeting requests, and then if the company is granted the MIDD meeting, paired meeting, then you would get a meeting date in the that quarter. So if it is Q3, then you are likely to get a meeting request perhaps towards the latter part of that quarter. So in my example, Q3, uh, a meeting estimated meeting date could be late October. Sometimes it could be early November as well. I hope that answers the question. Other questions? Okay, I do have, and if the attendees have thoughts, some of you have regulatory background. I know. Please do chime in. Um, the the would would the um, would this, it's an MIDD meeting initiative is a new program. I mean, relatively speaking, and from the from the publications coming out of FDA as well as the public talks, and the recent creation of the Center of Excellence for Quantitative Medicine, there seems to be this initiative seems to have gained a lot of traction, and in given this, from a pharmaceutical company point of view, Joan, relative to, let us say, an end of phase 2 meeting, which is almost considered a religious practice, meaning every program goes through that, and pre-NDA, pre-BLA meetings. Would this be considered a exotic meeting, meaning a resource intense meeting? So you would think, or what would be the considerations to, you know, to decide if MIDD would be a way to go? Is the resource intensiveness a consideration, meaning in terms of time commitment and such?
I think we're learning about the program. Certainly, talking to the individuals who participated in the pilot program between 2018 and 2022, I think there was generally positivity. It was a good experience, that it was very helpful in terms of the preparation work was good for the sponsor because it helped them organize their thoughts. It also is good for the regulatory authority because then they get a sense of what are the questions that are coming along and where are people getting hung up. I know that all of this is also important internationally. The ICH M15 is obviously paying very close attention to what is happening in these interactions. Now, how do we make the first step? Well, I think, of course, if a company, if a sponsor is interested in population pharmacokinetics, if they already embrace exposure response, I think all of the individual elements that make up MIDD are to more certainly most, I think most companies have an awareness that these tools are available. And they also, if they've had positive experience, that they have seen that they can get information in the label that they wouldn't otherwise have. They've got pediatric dosing that they wouldn't have. They've got, an estimate of adverse reactions or something like that. If they have been able to use modeling in some effective way that has saved them either time or treasure, they are much more likely to go along. I think it's the organization, the bringing together. It's not yet a rite of passage, but I think, if 62 companies have already had that experience and we double or triple that over the next 5 years for the interaction, we may be in a circumstance where essentially everybody has had some experience. And a sponsor only needs one experience that they can spread it throughout their examples. They can learn from it, but it strictly applies to one asset. So I think most companies will find it to be worthwhile. And certainly once they've had positive experience or learned from others what a positive experience it is, that it will become a rite of passage.
Thank you. And um, and the deliberations, considerations in terms of availing the MIDD meeting pathway, do you think, John, the considerations would be different between a small company, which may or may not have an army of clinical pharmacologists, versus a large company? The reason I ask is that the small companies, just looking around these days, are coming up with very novel therapeutics, which may or may not have direct policies in place. And MIDD could be a good opportunity to engage with the agency on innovative approaches. I just wanted to see if you had any thoughts, based on your experience.
I think it's been my experience that small companies, for the most part, are made up of individuals who have experience from other companies, and as well that they are well connected. But small companies understand that they need to use their resources very judiciously in order to push ahead. And innovative in therapy, innovative in approaches. And MIDD is, again, an opportunity to bring their particular asset to the attention of the regulatory authorities. Again, it's not that we have to have the answers for everything, but we have to have mapped out where we anticipate getting the responses. And I think if we're working in oncology, the difference between an MIDD meeting and an oncology dose optimization meeting is it's just a matter of semantics. I think regulatory authorities are going to continue to ask us to answer questions about MIDD, either as a MIDD paired meeting or in another meeting context. So I think it behooves all of us to pay attention to this opportunity and learn how to do it right.
Thank you. All right. And if I will keep going, and if there are questions, please don't hesitate to pose it in the chat box. Looks like we have a shy set of attendees today. Um, Now, I just want to have a brief plug-in for Wipro, just for those people who are not familiar with it. It's a drug development regulatory strategy themed company, and we do have experts who can support MIDD type activities and clinical pharmacology related and broad clinical development strategies. So feel free to contact us if you think we can be of help. I wanted to also announce the next regulatory briefs in our webinar series, which is pushing the 505(b)(2) envelope. And I would be presenting on this topic. Let me give you a brief rationale for picking this topic. 55B2, in my opinion, is one of the least clear areas in terms of policy from a regulatory point of view. There is a lot of flexibility, and that allows both the sponsors as well as the regulatory authorities to be innovative. And I would like to present how, in several instances, that 505B2 pathway um, was what led to unprecedented actions, regulatory actions. So please join us for the webinar. I think you are supposed to take a picture of that barcode. You will receive email as well about this announcement. So please note your mark your calendars. And finally, um, this is my final slide, but there is a poll, I think, that you would see in your chat box asking for feedback. It's just one question and one click from your side. If you can take a few minutes, please, to give your feedback, then we'll see if we are smoking something to keep doing these regulatory briefings, or we should be doing something else. Thank you very much.